Moving on then to the second half of this, really, by moving on to looking at blood. So there are four main components of blood that we're going to be talking about, one of which we're going to break down into two. But the first is red blood cells, which do give blood its characteristic red colour. We've got the white blood cells, which there are two different types, which we will talk about. We've got platelets, which you can see are smaller than even the red blood cells because they're not cells, they are cell fragments. And then finally, the liquid is the plasma. Okay, quite a lot to say for all of these different cell types, but the overall function of the red blood cells is transporting oxygen from the lungs to respiring tissues. There are a lot of adaptations that contribute towards the red blood cells being really excellent at transporting oxygen from the lungs to respiring tissues. One of which is this unusual shape. So we describe it as being a biconcave disc because it goes in on two sides. This does mean that relative to the volume of a red blood cell, that it does have a reasonably large surface area. Red blood cells are also quite thin because of this idea of them being disc-shaped. So they are thin and they are flexible. They can be kind of folded over. That's really helpful for being able to squeeze through the capillaries, which we talked about just a little bit earlier. The capillaries are the lumen is only the diameter of one red blood cell. So it helps for the red blood cells to be flexible so that you don't get blockages. Red blood cells. One of the key components then is hemoglobin, which is a protein. It's a red pigment. It's the hemoglobin that gives blood and the red blood cells their red colour. And it's the hemoglobin that binds to oxygen. So it's the hemoglobin that's actually transporting the oxygen around. The red blood cell itself is just a vessel containing hemoglobin. Another bonus to the red blood cell being very thin and having a very large surface area is that the hemoglobin is generally always pretty close to the surface of the red blood cell. So oxygen molecules don't have to travel very far to get from being outside of the red blood cell to being able to bind to a hemoglobin molecule. And that large surface area is going to contribute as well towards being able to take in as much oxygen by diffusion as possible into the red blood cell. In terms of the diameter of red blood cells, it's about 7 micrometers, which is very narrow. As I've said a couple of times, it's the same as the diameter of the lumen of the capillary, but they are one of our smallest cells, red blood cells. And just one more thing just to add for the red blood cells is that they don't have a nucleus. So red blood cells don't have a nucleus, and that means really just that the red blood cells can fit even more hemoglobin, which means they can transport around even more oxygen. It does mean that the lifespan of a red blood cell is limited because of the lack of a nucleus, but more room for more hemoglobin is more important. Moving on to white blood cells. So as I said, there's two types of white blood cells. The first is the phagocytes, which we can see on the picture there. They've got a really unusual shape. Particularly the shape of the nucleus is really unusual. So white blood cells, both of them, it's all about defending the body against pathogens. Pathogens, if you're unfamiliar with that one, is a word that means disease-causing microorganism. So that might mean a bacteria, it might mean a fungus, it might mean some kind of protozoa as well. It might be referring to a virus as well. So anything like that, any disease-causing microorganism, the word we use is pathogens. So phagocytes, they defend the body against pathogens by a two-part process, by engulfing and then digesting the pathogen. So because of that unusual shaped nucleus, is that phagocytes are able to manipulate the shape of their cytoplasm so that they can kind of engulf and move around the pathogen. And we describe the nucleus as being lobed in shape. Also allows the phagocytes to be able to squeeze out through the capillaries and enter into the tissues as well. And then once they've engulfed a pathogen, is that they need to be able to digest it. So they'll do this using enzymes. So they'll use enzymes to break down the pathogen, and they'll either absorb and use the parts of the digested pathogen, or it might just be left as waste. The way that phagocytes do this is it's non-specific. So a phagocyte could engulf and digest a fungi, a bacteria. It doesn't have to be the same species. It doesn't matter. So it's a non-specific response, which is quite different from lymphocytes, which is our second type of white blood cells. These are really specific, and they release these specific antibodies. So antibodies are a protein in response to a specific pathogen, and in response to the antigens, which are a structure on the outside of a pathogen. So in response to a specific antigen as well. So in the same way as where we've talked before about enzymes and substrates being specific, it's that same kind of specificity. You'll have to forgive my artistic skills here on this one, but just to get a bit of an idea of what these different terms are referring to. So this structure that I've drawn would be the pathogen, and then I've just drawn one antigen on the outside of the pathogen. There would be more than one. I've just drawn one just for this illustration. And then that next structure that I've drawn would be an antibody. Now you can see there, even with my drawing, that the antibody is a complementary shape. Okay, so that term hopefully is familiar from enzymes, that it's a complementary shape to the antigen. So the lymphocytes are going to be producing and releasing specific antibodies that are going to be that perfect complementary fit to the antigen of a pathogen. And by doing that, they're going to cause the destruction of the pathogen, either by destroying the pathogen directly or by functioning as a kind of tagging system, so that the phagocytes, that other type of white blood cell, can find this pathogen a little bit more easily. So there I've written there, so antibodies can kill the pathogen. Obviously by using that word kill is that you could only really use that sentence to refer to something that was alive to start with. So you've got to be quite careful if the specific pathogen you're referring to is a virus, because viruses aren't alive to start with. They can't be killed by antibodies. But if we were referring to a bacteria or a fungi, then you could definitely use the word kill. Destroy, or causing the destruction of, is often a safer term to use instead. Okay, platelets then. So as I said before, they are cell fragments, and they are absolutely vital to blood clotting, which is really important if you become injured, if one of your blood vessels becomes ruptured, so that you don't lose lots of blood, is that the platelets can cause a blood clot to form. They do this by releasing chemicals. They do this in response to coming in contact with tissues outside of the blood vessel. And then they cause a soluble protein, which is called fibrinogen. And that fibrinogen would be found in the blood plasma the whole time anyway. It's normally found there. And so that soluble fibrinogen would be converted to insoluble fibrin. And in doing that, it all starts to stick together, and it forms a mesh. And that mesh will start to trap platelets. It will start to trap red blood cells, white blood cells, and it will start to form a clot, a clump of cells all together, which if that, once it dries out, will form what we call a scab as well. And just thinking quickly about why this is so important. So it's really important for blood vessels to clot if they are ruptured, because otherwise your body would be really vulnerable to pathogens entering your bloodstream. So by causing clotting, by blocking entry, that they're preventing entry of pathogens to your blood, but also making sure that you don't lose lots of blood. Okay, our fourth and final component of the blood then is the blood plasma. So this is mostly water. It's a straw-coloured liquid, so a kind of pale yellow colour, because it gets that colour from some of the different things that are dissolved in the plasma, but as well as being a transport medium for the cells and the platelets, is that it's also going to be transporting around loads and loads of dissolved chemicals. Specific examples include carbon dioxide, urea, glucose, and hormones as well. There are other things as well, but that's just a few examples. Because blood plasma is mostly water, it does also contribute to distributing heat around the body, which is often commonly overlooked. Looking at this summary then of red blood cells, so the presence of hemoglobin is going to allow for the binding of oxygen. When hemoglobin binds to oxygen, it forms something called oxyhemoglobin. Because of red blood cells' small size, they can fit through very narrow capillaries. And because the capillaries are so narrow, is that red blood cells do have to travel very slowly, which, as I said earlier, means that there is loads of opportunity for exchange. Having no nucleus means that there's more space for hemoglobin, which more hemoglobin means transporting more oxygen. And then because there's so many red blood cells, it means that your body can constantly be transporting around lots of oxygen to lots of different places. Not every red blood cell is going to make a journey around to all of your different organs. They're all going to different places, back and forth between the heart, the lungs, the heart, the body, etc. So you've got to have loads and loads of red blood cells. And this picture here just shows the kind of binding between oxygen and hemoglobin in the lungs, transporting that around in the red blood cell, and then the unbinding of oxygen and hemoglobin in the muscles. So oxygen and hemoglobin will bind together where there's lots of oxygen available, such as in the lungs, and oxygen and hemoglobin will unbind when there's not very much oxygen around, such as in respiring tissues. Okay, looking at these final questions then, do make sure that you've had a go at these before watching through this, because I will just be filling in the answers here. So cell A is a white blood cell, specifically a phagocyte, identifiable because of the lobed nucleus. B is going to be a red blood cell, identifiable because you've got the hint there of that biconcave disc structure. C is going to be producing antibodies. C is a lymphocyte. We can see that because it's got the round nucleus rather than the lobed nucleus. How do antibodies prevent infection? So they contribute to the destruction of pathogens either directly or by marking them and making it easier for the phagocytes to destroy them. And then we've got a question there asking about how phagocytes work and how phagocytosis works, so engulfing and digesting the pathogen. And that will obviously take that pathogen out of circulation in your body so that it cannot release toxins and it cannot damage the body. And then how do platelets contribute to preventing infection? So they're going to both prevent entry of pathogens to your body and also reduce potential blood loss from your body by forming blood clotting in response to injury.